Hello, APBIO. In this video, I'm going to try to put everything together to think about, we've been talking about all this control of gene expression, and the purpose of control of gene expression is so that we can have cell specialization. But I want to talk about it now from the viewpoint of cell specialization. So what does cell specialization look like? And the cell specialization starts with cells that have not been specialized, cells that could be anything. And we call those totipotent stem cells. You've heard of stem cells before, probably. Totipotent stem cell. And that's just an undifferentiated cell, a cell that can become any type of cell. Now, when there are genes that are turned on, some of those genes are just regulatory genes, different developmental genes that determine what a cell is going to be. And we call that the process of determination. And determination are just these progressive restrictive steps in the developmental potential of this activation of these specific regulatory genes. So they're genes that are saying, okay, you can now become a muscle cell or you can become part of the nervous system, but you can't, once you're determined, you can't switch back and forth between systems. So it's not yet a completely specialized cell, but it's determined what it's going to, what type of cell it will become. So there are progressive restrictions in developmental potential of master regulatory genes. And so some of the things that can happen are certain genes become a part of the heterochromatin, the chromatin that's packed really tightly, where the DNA is methylated and it's all scrunched up tight. And those genes can't ever be accessed. So those pages are permanently closed in that part of the genome for those types of cells. So a cell that's been determined, an example of a type of cell that's been determined, is something like a myoblast. Now, a myoblast can eventually become a muscle cell, but when it's a myoblast, it's not a specific type of muscle cell. It's just on its way to becoming a muscle cell. And now it's never going to become a skin cell. It can't. It's got too many restrictions on it. So some of those opportunities that were available during when it was a stem cell have now been restricted by these master regulatory genes. And then following determination, we get cell differentiation. And cell differentiation refers to the specific processes that result in a specialized cells. Process So now we have like a muscle cell and even a specific muscle cell. And that muscle cell, it can't ever become any other type of cell because it's been restricted to the point where it will only be that type of cell. So in that way, you can see what cell specialization, what the effect of all of controlling of the expression first controls in general what the cell can become with cell determination. So at first, stem cells can become any type of cell. And then in general, there's restrictions. Master regulatory genes are going to put restrictions on cells. And then finally, cells become specialized to a particular type of cell. Let's look at another path of cell specialization, sort of a specific example that is when a single cell, a zygote, mitotically divides but becomes all these different types of cells. So that's embryonic development. So this is the process of you developing, or any organism developing from a single cell to a multicellular organism. So process of that single cell, remember we call that a zygote, and that develops or developing into a multicellular cellular organism. And the divisions that are happening are mitotic divisions. So when we thinking back to mitosis, when we talked about mitosis in Unit 5, remember that mitosis produces two daughter cells that are genetically identical to the parent cell. But the mitotic divisions in embryonic development actually produce different cells. And let's talk about why. The mitotic divisions produce different cells as a result of either things that are coming from the inside of the cell, factors, environmental factors or substances inside the cell, but then also outside the cell. So some of them are inside, some of the factors are inside, and some of the factors are outside. One of the internal factors that results in these different types of cells are substances called cytoplasmic determinants. And cytoplasmic determinants are substances that come in the cytoplasm from the gametes. And because the cytoplasm all comes from the maternal gamete, we say they're also called maternal substances. And there are things like RNAs and different proteins that exist in a variety of concentration gradients. So if you think about what that means, hold on, let's write that down. They exist in a variety of concentration gradients. And then what happens is, if you think about the way they divide, you can see this diagram right here. When they divide, there is, if this is a single cell right here and it hasn't divided yet, if there's more of a particular substance, let's say this blue substance, toward one end, one pole of the cell, and less of it towards the other pole of the cells are actually different. So you get it leads to unequal cell division. And it's just because of all this stuff that's inside. When the cytoplasm divides, you get more of a particular protein, and it's all very tightly controlled. The substances that make up cytoplasmic determinants, they actually control gene expression. So there are different things that are going to control the expression of genes along the central dogma. So they could be different interfering RNAs. So they interfere with whether a pre-translational modification, or there are different proteins that are already there that can turn different processes on, different transcription factors that can turn processes on. So the cytoplasmic determinants specifically are substances that control gene expression, and so therefore they lead to different cells, different types of cells, because they're controlling expression. So that when that embryo is dividing, that zygote is dividing here, and we have some cells becoming one type of cell and then some cells becoming another type of cell. And as the blastosphere forms, you get actually layers of tissues. You have all the different layers, 3 layers of tissues in higher animals, And because of that, you get different systems. Like, for example, the middle layer becomes your digestive tract, and outer layer becomes your integumentary system, and so on and so forth. So you get these different cells, and the cells are first they all start out as stem cells, totipotent cells, but there's progressive restrictions because of these restrictions, because of these presence of cytoplasmic determinants, and then you get cells that are determined, and then eventually you get cells that are differentiated. Now, there's also external signals. Now, not necessarily external from the organism where it's developing, but just external from the cells. And here we have the process of induction, and we talked about this way back in Unit 4. Induction is just adjacent cells are going to communicate to foreign tissues. And if we look at here in this diagram here, you can see here's these two cells in this picture, this cell right here and this cell right here are talking to each other. And this could be a cytoplasmic determinant right here, but it's going to release it into the extracellular space, and it's telling this cell, hey, let's be a limb bud. Let's become part of the digestive tract. Let's become this type of tissue. Remember, a tissue means all cells that have the same function that come together, that all the same cells that come together to sort of like teamwork to perform the same function. Anyway, induction is responsible, and actually this cytoplasmic determinants and then the process of induction, those two things together are responsible for establishing the body axes. So there are three main body axes in all bilaterally symmetrical organisms. Bilaterally symmetrical organisms, of course, have a left-right. But then if you think about your body, you have also a dorsal ventral. Dorsal is your back. Ventral is your chest. So all bilaterally symmetrical organisms have that. And then they also have the front to back. That's the anterior, posterior. So the three body axes are established by the cytoplasmic determinants and this process of induction together, which means that your mother put your head between your shoulders and your fanny behind you. And that's good that those things are in the right place. Okay, I hope that was helpful. You can see how cell specialization begins as soon as that first cell is made from the two gametes. Cell specialization begins in the zygote with the formation of concentration gradients of cytoplasmic determinants, and then those cytoplasmic determinants are going to be used for communication to form tissues, and so on and so forth. So we get a series of mitotic divisions that result in unequal cells, and that's how we have multicellular organisms that can be that are generated from a single cell through a many series of really divisions that are genetically identical, but you have all different cells, or all multicellular organisms are composed of many different types of cells. Okay, that's it, and I hope that was helpful.